No, thank you so much for your presence today. Nice to meet you too again. Bueno, buenas tardes y buenas noches también aquí en Brasil. Buenas tardes para la mayoría de los países en Latinoamérica. Y de repente también hay otras personas que se conectan desde Europa o desde Asia. Bienvenidos también. Bienvenidos. Welcome for everyone. Vamos a empezar esta presentación, este webinar. Voy a volverles a compartir mi pantalla para que ustedes puedan tener una visualización general de lo que significa nuestro evento. Ok. Un segundo. Ya. Un momento. Have some problems to share my screen. Ok. Ustedes me pueden confirmar si pueden ver mi pantalla. Ok. ¿Me pueden confirmar, por favor?
Sí, Clary.
¿Me pueden confirmar que solo ven mi pantalla también?
Sí.
Ya, perfecto. Ok, vamos a empezar hoy día con esta presentación. Y voy a darle algunas informaciones importantes. A ver, un momento. Listo. Bueno, buenas tardes a todos. Como ya les había saludado, quiero darles la bienvenida a este evento, Bioinformatics Applications. Y en este día tenemos nuestra tercera edición, nuestro tercer webinar, en la temática de biología estructural, que lleva como título How to Extract Biology from Protein Structures, en nombre de la Sociedad Peruana de Bioinformática y la comunidad científica lambayecana. Mi nombre es Cleidio Osorio, my name is Cleidio Osorio, y soy parte del comité directivo de la Sociedad Peruana de Bioinformática y seré su moderadora en el día de hoy. Bien, la Sociedad Peruana de Bioinformática es una asociación peruana sin fines de lucro, fundada el 23 de julio del año 2019, que tiene como objetivo integrar a la comunidad peruana dedicada a la bioinformática, impulsar también el uso y desarrollo de la bioinformática en el país, en este caso Perú, combatir las disparidades y promover las buenas prácticas en investigación. Y la comunidad científica lambayecana, que también es organizadora de este evento, es una comunidad de científicos lambayecanos. Para los que no saben, Lambayeque is a city in the north of Lima. The capital of Peru is Lima. In the north of Lima is Lambayeque, is a department. There are a lot of scientists there too. Y esta comunidad de científicos tiene un objetivo: reunir también, contactar, organizar a profesionales lambayecanos quienes se desarrollan en diferentes áreas de investigación en el ámbito nacional y también internacional. Y de esta forma, esta comunidad buscaría tener representatividad en temas de interés tanto regional como nacional. Muy bien, en esta oportunidad tenemos al doctor Richar Charles Garrat. Voy a hablar un poco más sobre él. El profesor Richar es profesor titular de la Universidad de Sao Paulo, Brasil. He's a professor in the Sao Paulo University, Brasil. Él tiene experiencia en el área de biología estructural, utilizando técnicas de cristalografía de proteínas y modelamiento molecular. In general, this is about biology structure, protein crystallography, and molecular modeling of proteins. Sus áreas de investigación actualmente incluyen septinas, superóxido dismutasas, la vía de rescate de purinas en parásitos, receptores nucleares, diseño de fármacos y vacunas basado en estructura proteica, ingeniería de proteínas y en el desarrollo de herramientas didácticas para la enseñanza en biología estructural. So he worked with parasites, worked with receptors of the nucleus, and design of drugs and vaccine, based in protein structure. Y él también es miembro de la Academia de Ciencias del Estado de Sao Paulo y de la Academia de Ciencias Brasileña. ¿Ok? Muy bien, voy a explicar ahora la dinámica de las preguntas. Bueno, cuando se finalice la presentación, que será aproximadamente de 30 minutos, se va a proceder con las preguntas que alguno de ustedes, los asistentes, pueda tener. Así que les agradecería que puedan dejar sus preguntas en el chat de esta plataforma Zoom y yo los voy a leer, voy a proceder a leerlos al final de la presentación. Pero también hay una opción que si desean, pueden hacer sus preguntas directamente al profesor, terminando obviamente la presentación, encendiendo sus micrófonos. Muy bien, entonces agradezco a todos los asistentes. Apaguen sus micrófonos y las cámaras para evitar interrupciones. Well, in the end of the presentation, you can, I'm going to say your questions. You can leave your questions in the chat box of this platform, Zoom, or you can on your microphone to ask to the doctor in the end of the presentation, please. Please, and now in this presentation, off your microphone, your phones, and your cameras to avoid interruptions. Ok, sin más que informar, procedo a cederle la palabra al profesor Dr. Richard Charles Garrat. Please, Doctor, this is your time of your presentations. Thank you so much for your presence.
Thank you. Muchas gracias. Buenas noches. Lamentablemente, yo no hablo español, entonces I'm going to speak in English. Okay. But I'm sure that we should be able to work this out together when we get to the questions. Now, what do I have to do here? I have to share my screen. Okay. Now, can you now see my screen? Can you see that?
I can see.
Yes, you can see. Okay. So we assume everybody can see it then. Okay. So this is the title of my talk. How to extract biology from protein structures. Okay. So as Clady very kindly pointed out, I am a structural biologist, a protein crystallographer. We solve structures. Actually, before I go any further, let me thank also Percy for the invitation and Marco for setting up the technological aspects of this Zoom webinar. Okay, so that's the thank yous. Now what am I going to talk about? Well, extracting biology from protein structures. What do I mean by that? What I mean is, when we solve a new structure of a protein, normally.
Excuse me, dear doctor, we can see the screen, but just a color. Real is not a presentation.
Oh, sorry, what are you seeing?
We can't see the presentation.
Oh, you can't see the presentation. You can see my, what are you seeing?
We can't see. We can see your screen of your PC, but we can see the presentation.
Oh, please move this window away. I don't, okay. Anybody know what I have to do to make you see this presentation? I did this yesterday and it worked.
It is a problem. It's common, really.
Okay, let me.
Na verdade, é um problema muito comum. Eu vejo como cinza.
I'll say software cinza. Okay. So if I do this, let me try this.
You can try again, please.
Yeah.
Okay, now we can see.
Now you can see it.
Okay. Problem. It's a common problem.
So I was sharing the wrong thing, obviously. I apologize for that. Okay. So can you now see it big? Yeah, is that okay?
We can see now. Good.
You can see it. Okay. That's
Good.
good. Oh, well, I can't see it. Why can't I see it? It's gone away. Okay. Oh dear, oh dear. So you're seeing that okay?
Sim.
Clady, are you seeing that?
Yes.
Yes.
Mhm.
Okay. Well,
But
let's start.
now I can see you, your
Good.
face.
Oh, you can see my face. You can't see the slide?
Yes.
Oh,
Your
dear.
slide now.
So what's happening here? Because
No, but I could see your slide.
you could see?
A few 50 seconds.
Oh, okay.
But now I
Host
suppose
disabled
you have to share again.
It's giving me a message here. Okay, host disabled participant screen sharing. Oh, I don't know what that means. Okay.
Okay. You can try again, please.
Yeah, I'm going to try again. I don't know.
Eu acho que você compartilhou seus slides, faz
Yeah.
um minuto e depois foi embora.
I've lost I can't even see the green button now. I don't know what's happened. I think I'm going to have to leave and come in again. Or what does that do? I can't see. I don't even get the green button now.
You can try to share again. Just
okay, leave.
Alguien menciona en el chat que a lo mejor es necesario que le den permiso para compartir la pantalla.
No, ya tenía permiso. Él tiene el permiso ya, porque hace un momento pudo hacerlo. Pero de todas maneras ya di el permiso.
Mejor que vuelva a entrar porque Zoom ha tenido estos problemas últimamente.
Listo.
Is that working? Can you see my screen?
We can see, doctor.
Okay.
Now.
Is that okay?
Okay.
Okay. Let's go with this. Can I
Thank
start?
you so much. Thank you so much.
Right. Okay, so the problem really is how to look at a new structure and discover what the structure is trying to tell you. I mean, what is the biology? How does it work? How does the protein function? Most of us teachers tell students that there's an intimate relationship between structure and function. And that gives the impression that if we solve a new structure, then it will be obvious how the protein works. But that's not really the case. It never has been the case. So for example, these guys here, this is Max Perutz, when he solved the structure of hemoglobin, looking at it, three intelligent old gentlemen trying to work out how does this thing work. And the problem, it's really a problem of reverse engineering. It's not that we've designed the protein. If we designed the protein, we'd know how it works. It's reverse engineering. It's as if we discovered a crashed flying saucer and we have to work out how does the anti-gravity device work. So it's the opposite of a problem of design. Now, design is easier. For example, this beetle has a problem with temperature. It lives in a cold environment. It needs an antiphase protein, which will prevent ice crystals from growing inside its cells. This is the structure of an ice crystal. So how can you design a protein which will prevent an ice crystal from growing? Well, there are proteins which have flat surfaces, an array of hydroxyl groups on them, which match the crystal lattice of an ice crystal and will sit on the surface of small ice crystals, preventing them from growing. So if we were to solve the structure of this molecule and we knew what its function was, it would be easy because we've designed it to do something. The problem is, when we solve new structures, nobody designed them. They are the result of evolution, of natural selection. So we have to look at them and try and interpret how they must work. That's not easy. So there's several different things we could try to get out of a structure. For example, what does my protein do? Well, normally we know what it does. Before we have a structure, we know what it does. And we crystallized it to solve its structure because it was an interesting thing to study. Where does it do it on the surface of the protein? That is to say, where is the active site of the protein? It's a second kind of question. And how does it do it? That is to say, what is the mechanism by which it does it? So if it's an enzyme, what's the reaction it catalyzes? Where is the active site? And what's the mechanism it uses, the catalytic mechanism? Now, normally we know the answer to the first one. We normally know what reaction it catalyzes. So before we have the structure, we don't know where it does it on the protein, and we don't know how it does it. So these are questions we want to answer. So there's a few general thoughts about how to go about doing this, okay? So the first general rule is that there are no general rules, okay? But don't overinterpret the structure. There's a very important number in X-ray crystallography called the resolution of the structure, and that gives you an idea of the precision that you have in the atomic coordinates. So don't overinterpret your structure, okay? Normally, you can't get all the answers just by looking at the structure, so you have to know the literature. There is lots of software available. You guys who are bioinformaticians, I'm not a bioinformatician, or at least I don't consider myself to be a bioinformatician, but there's lots of software created by intelligent people to help you find interesting things in your structure. But it will often depend on your own insight and knowledge. Knowing the system well that you're studying is useful. And knowing something about protein structure in general is very useful. For example, if I show you this structure here, probably many of you who are used to looking at protein structures and reading biochemistry textbooks will recognize this as being a tin barrel. And it's got 8 beta sheets strands in the middle and 8 alpha helices going around the outside. So when you look at that, you immediately recognize it as what it is. But if I show you some of these things, are you capable of recognizing that there's something strange about these structures? This structure over here, it's got helices down here, it's got beta strands here, but what is this strange thing here? It turns out to be a very unusual structure, and that very unusual structure is almost certainly trying to tell you something about this, how this protein works. We won't go into the details. This is the example of the heat shock protein. It has a very flat surface. Most proteins are knobbly and irregular. They don't have flat surfaces. If you find a protein with a flat surface, with a regular array of hydroxyl groups, that's trying to tell you something. But you will only know that if you know well protein structure, if you're able to recognize these things, or if you have some clever software available which will analyze the structure and pick out the interesting things. Here's another one where you have a buried lysine residue, which is very unusual. It's probably trying to tell you something. Well, actually, I lied to you. When I showed you the tin barrel, I showed you this structure here. But in fact, that's not the correct structure. All of the tin barrels you've ever seen actually look like this. This is the mirror image of this. If you ever find a tin barrel that looks like this, please send me an email, because I'd be fascinated to look at it. It's almost certainly wrong, because there's good reasons why a tin barrel should look like this. You see how the strands are inclined like this and not like this? That has to do, at the end of the day, with the fact that protein structures are made of L amino acids. And if you follow the rules of protein folding, you inevitably come to the conclusion that the tin barrel must fold up this way and not this way. So knowing something about protein structure is extremely useful. Use as much information as you have. In protein crystallography, what we actually determine is an electron density map of the of a part of the unit cell of the crystal. So you'll have the electron density of the protein, and often there's extra electron density, which isn't protein, and that can tell you something. For example, look at this fantastic molecule here, which was found in a crystal structure unexpectedly. That told the authors that this had something to do with the synthesis of diazole, because this is an intermediate in the metabolic pathway which leads to diazole. So that's an extremely useful piece of information. This is just a fatty acid. Actually, it's a PEG molecule, looks like a fatty acid, and it's telling you something about how fatty acid binds to this phospholipase. Sometimes you can see a sulfate ion. That's very common because we often use sulfate to crystallize proteins. And the sulfate ion often occupies a position normally occupied by a phosphate ion, which is very common in biochemistry. And that may be telling you something about where your substrate binds to your enzyme. Okay, use as much information as possible. So we should look at proteins at their primary structural level, secondary structural, tertiary structural, and quaternary structural level. So can we get anything just by looking at the quaternary structure? That is to say, basically the symmetry which relates more than one subunit in an oligomeric protein. So there are very many reasons why many proteins are oligomeric. In fact, most proteins are oligomeric. Only about 20% of proteins are actually monomeric. So most proteins are oligomeric. Why are they oligomeric? Well, there's many reasons for being oligomeric. It may have to do with cross-linking, as in the case of antibodies. So you have to have more than one binding site. It may have to do with cooperativity and allosterism. It may have to do with forming cavities, like in ferritin, which stores iron, or channels, which go through membranes. It may have something to do with just being big and stable. So there's many reasons why you might want to be oligomeric, but just knowing that the protein is oligomeric and what symmetry it has, does that really help you? Not very much. I mean, there is some information there, but not much. For example, here's a table classifying oligomers according to their symmetry. This is the potassium channel. You can see it has a little hole, and it's very tightly regulated, this hole, so that the potassium channel is able to transport potassium ions, and it's so sensitive, it's able to distinguish a potassium ion from a sodium ion. So clearly, the arrangement here of the 4 different subjects to make that channel is very important. The symmetry of the molecules related to its function. This is another one which has a bigger channel. That's a hexamer, and that transports proteins, protein secretory apparatus. So the channel must be bigger because it's excreting a larger molecule. So there's some relationship between function and the size of the oligomer, but it's very weak information. There's not very much useful information just by looking at proteins at their quaternary structure. And they can be pretty tricky to work out anyway, because in crystallography, what you will find deposited in the PDB, the protein data bank, which has all of the coordinates of all of the crystal structures solved, is something called the asymmetric unit. That's the basic unit in a crystal. But that doesn't necessarily correspond to the biological unit. So in this particular case, we know that this molecule is a dimer, but in the asymmetric unit, there's only a monomer. And this is what the crystal unit cell looks like, all of these 8 monomers over here. So there's several different dimers which you can make from this monomer using this unit cell. It could be this one, could be this one, could be this one. And the software PISA is a very powerful piece of software which will look at the interactions between these different monomers in these different dimers, and it will tell you which is the most stable, which is the most likely one. Shape complementarity between the 2 components is also a measurable quantity and can be useful in distinguishing between a real dimer and an interface which is not a real dimer, it's just a crystallographic compound. But sometimes even that's not good enough. Sometimes you need a bit of experimental information. So here's an example of an enzyme where there was doubt about whether it was one kind of dimer here called dimer 1 or another kind, dimer 2. Both of them were possible from the crystal packing. The researchers knew it was a dimer, but they didn't know which dimer it was. So they solved it experimentally, not computationally, by doing a small angle X-ray scattering experiment, which are these little circles here. And then they theoretically calculated dimer 1, dimer 2, and a monomer. And clearly, dimer 1 is the right answer because the solid line fits the experimental data very well. So this can be done normally, computationally, just by looking at the interfaces, but sometimes you need a bit of help from experiment. So what about 3-dimensional structure? Well, A few years, so I'm now going to tertiary structure, not quaternary structure, tertiary structure. That's the fold of the molecule. Can we get any information just by knowing what sort of fold our molecule has? Well, this is just a little table that Christina Rengo and I developed a few years ago. It's called the protein chart, and it's a summary of all of the PDB on one piece of paper, okay? It's divided into alpha proteins, beta proteins, alpha, beta proteins. And around each example of a fold, there's a little octagon here. And the octagon has little colored bars, which are associated with different kinds of function. So the question is, if I know, for example, that my protein has the fold of a beta trefoil, and once I've solved the structure, that's easy, okay? So I can work out what fold it is. But does the fold tell me what kind of function it is and how it does it? Well, you can sometimes get some low resolution information, but not often. A classic example is the Tim barrel. So this is the Tim barrel fold. It's very much associated with this blue bar, which means enzyme activity. Almost all Tim barrels are enzymes. There are exceptions, but almost all of them are enzymes. So if you solve a new structure and you discover it's a Tim barrel, you not only know that it's an enzyme, but as I'll show you in a minute, we can work out where the active site will be. So being able to compare a new crystal structure with another, with a bank of crystal structures is useful because it can give us hints about what the structure does. For example, here are two proteins which initially look very different. This one's much bigger, much smaller, but if you look at the part in black, they have the same fold. And so if you solved this structure and knew what the function of this one was, you would be able to have a good guess, not about just what it does, but where the active site in this enzyme is. It turns out that both of these are called P-loop hydrolases. So they bind an ATP or an ATP-like molecule involved in hydrolysis. Okay, so sometimes, yes, by getting a fold, you can work out something about what your protein might do. But this slide just shows how the correlation is not very good. Here on each one of these circles, green means beta sheet proteins, red means alpha helical proteins, and yellow means alpha beta proteins. And you can see for these different functions, heme binding, carbohydrate binding, nucleotide binding, DNA binding, you have examples of all three of them. It's true that if it's a nucleotide binding protein, it's probably going to have a fold, which we call alpha beta. So it's got both alpha helices and beta sheet. It's probably going to have that kind of a fold, but there are examples which are based on alpha helices and others based on beta sheet. So once again, I call this low resolution information. Just because I know that my protein fold up in a particular way doesn't really tell me the details about what it does, which is what I would like to know. So how do we go a bit further, look a bit more in detail? Well, by comparing in more detail, not just the fold, but how well a new protein structure superimposes on known protein structures, then it's possible. This program 3D Fun is an example which tries to do this. And what it tries to do is go down within the enzyme commission code classification, which is 4 numbers separated by dots. So this is 3.12.2.1, okay? And the first number tells you what general class of enzyme it is, like a lyase or a ligase or whatever. And it gets more and more specific. By looking how closely this new protein resembles average proteins within each of these different categories, it's possible to speculate about what its function might be. So for example, this particular protein was solved in a structural genomics initiative, where they were just trying to solve as many structures as possible, so without worrying about what they did. And they looked using this method 3D Fun and were able to predict that it was probably an indolamine 3,2-dioxygenase. So now we're getting a bit more interesting. We're starting to say something a little bit more detailed about what my protein does. Another one is a structure called 3D ligand site. It does something similar. It superimposes structures. And if these structures have something bound to them, a ligand bound to them, then it can look to see if that ligand ought to bind to the new structure also. So it'll tell you what the ligand is and where it will bind. Okay, so that's tertiary structure. What about secondary structure and motifs present in protein structures, which are composed of a small number of secondary structural elements? Can they give me any useful information? Well, for example, let's say I solve a new structure and it has an architecture which we call a doubly wound alpha beta alpha. Okay, what is that? Well, it's a 3-layered structure where you have a beta sheet in the middle of the structure and alpha helices on both sides. So it has 3 layers, alpha, beta, alpha. These molecules always have what we call a complex topology to the beta sheet. And that complex topology can be used to predict where the active site is. So if I solve a structure which has this kind of an architecture, a doubly wound alpha, beta, alpha, I don't know where the active site is, but just by looking at the topology of the beta sheet, what does that mean? By looking at the way in which the beta strands in the sheet are connected together, I can predict where the binding site will be. And it works like this. You have to find the place in the beta sheet where the following loops go to different sides of the sheet. That forms a cavity, which is perfect for binding my substrate or cofactor or whatever it is. Okay, so just by looking at the topology of the molecule, I can actually predict where the binding site will be. And in the case of tin barrels, I can do it also, because it turns out that all tin barrels, despite being besides the fact that they're enzymes, they all have the enzymes in the active site in the same place. It's all at the C-terminal end of the beta strand. Okay. So as soon as I solve my new structure, I see it's a tin barrel. Immediately I know, 1, it must be an enzyme if I didn't already know. 2, I know where the active site is, because all of active sites in tin barrels are always in the same place. So I'm starting to get some more useful information. So for example, here are 2 examples of architectures of proteins which have the alpha, beta, alpha 3-layered arrangement. These circles represent alpha helices and the triangles represent beta strands. So I can just look at this. I see that this one goes up, that one goes down, so I can predict that's where the binding site's going to be. And in this case, that one goes up, this one goes down, that's where the binding site. This rule works almost 100% of the time. So what I'm saying is, when I have a new structure, it is possible to look at it and detect where the binding site probably is. This is just another part of that chart I was talking about, which gives you lots of rules about lots of different motifs. I took one example there, which is this example, basically here. Okay, but the devil's in the details. To really understand function, we need to look even closer at the structure. We need to get down to atomic detail. Quaternary structure just tells me where the subunits are, one with respect to another. It doesn't tell me much. Tertiary structure tells me about the fold. It tells me something, it doesn't tell me much. I need to look at the atomic details. So visualization is very important. There's lots of software out there, the bioinformaticians probably know most of them, which enable me to draw protein structures in lots of different ways. So I can map them onto the gene structure and see where the exons are. Sometimes an exon is associated with a different function, a particular function, so it can be useful to notice that within my 3-dimensional structure. I can look at where sequences are conserved within a family, or how structures compare one with another within a family. That can tell me something about domain movements, how a protein may move in time and space. All of this information can be useful with respect to function. And I need to find where interesting things are in my molecule. So I need to be able to have different ways for visualizing how a ligand interacts with a protein. Often we get a lot of information from our crystal structures because we crystallize the proteins together with either an analog of the substrate or the product or an inhibitor or some other molecule which permits us to see the interactions, if it's an enzyme, for example, which we would expect to find between a substrate and an enzyme. But looking for cavities is extremely important. Why is looking for cavities important? Because a lot of active sites in enzymes, for example, are in cavities. It's not the same as a binding site on an antibody, which is going to be on the surface. But it's very important for many catalytic mechanisms to exclude water from the active site. So it's important that the active site be buried somewhat inside the structure. So software, which can look at my molecule and detect where I have a cavity, or a cavity or a groove, can be extremely useful for generating hypotheses. Looking more closely at the side chains is very important because we know that particular constellations of side chains are found in particular kinds of proteins. For example, there are many different kinds of serine proteases. They're all proteins. They all have a catalytic serine which hydrolyzes a substrate, which is also a peptide or a protein. But there are many groups which are not homologous. They're not related evolutionarily. They are the result of convergent evolution, but they've converged so that you have a serine, a histidine, and aspartic acid in the same arrangement based on three-dimensional structures which are otherwise completely different. So if we can look at a new structure and find constellations of side chains of this type, we ought to be able to predict function from structure. So some programs called, one program called Spasm, another one called Rigor, can do this kind of thing for you. Here's another list of more software which is out there available to look at the details of how the side chains are arranged in structures and try to say, this looks like an active site which will perform this kind of catalysis, or this is a copper binding site, or whatever. So here's some examples of cavity finding software. Actually, there's loads of software out there. You bioinformaticians are very innovative. I mean, you know, there always seems to be an infinite number of programs to calculate things. And that's no different in protein structure. So there's many softwares out there which will allow you to calculate cavities in proteins. And then you can look at those cavities and see if they look like they might be active sites. Okay, but what we really want is real detail. How could we get that? Well, here's an example. This structure was solved from structural genomics. So they solved the structure, no idea what it did. Okay, well, they knew it was an enzyme, but they didn't know what it catalyzed. They didn't know where it catalyzed it in the structure, and they didn't know how it catalyzed it. So they just had the structure. What can you do then? Well, they did a very clever study. They did a docking study where they docked into the probable active, first of all, find the cavity of the active site, and then they docked into the cavity or the likely cavities, not potential substrates, but potential transition state analogs, because these transition state molecules are more compatible with the active site structure. They're more complementary to the active site structure than the substrate itself. That's what induces catalysis to occur, as Linus Pauling pointed out. So they took a whole bunch of compounds which are known to be intermediate transition state analogs in biochemistry and tried to dock them all into the active site. And what they found was they got several hits where all of the analogs they used were adenine analogs which underwent C6 deamination. They had a scoring function sufficiently good to pull out all of these adenine analogs as the top scores. So it's telling you not only what kind of molecule it is, but what kind of reaction it does. So what did they do then? Well, they said, well, let's get one of these C6 deaminated adenine compounds, and we'll soak it into the crystal structure and see if it actually binds the way we're saying it binds. And believe it or not, this is the superposition of the crystal structure in red. This network here is the electron density. And the green molecule is that predicted by modeling. So this is amazing. This is telling you your molecule's an enzyme. It tells you what kind of substrate you have. It tells you what kind of reaction is being catalyzed, and all just from the crystal structure. Now, this is, of course, an exception. I mean, this is the example we use to demonstrate how this can be done. Most cases, this is very, very difficult. Okay. not all proteins are enzymes. So some proteins just interact with other proteins. So it's useful to be able to look at a new protein and see if we can predict where on its surface it's likely to interact with a binding partner. No proteins do anything very interesting without interacting with other molecules and just sit there isolated. Some of them bind small molecules, like enzymes do, but some of them interact with other proteins. They can be signaling molecules, or they can be molecules associated with the immune system, which recognize foreign components from an invading organism, proteins, DNA, whatever. This is one program, Sharp Squared, which will look at the protein surface of a new structure and will say, this is where I think something binds to it. This is just knowledge-based. It just analyzed all of the PDB effectively and come up with the characteristics that a binding site normally has. And there's lots of other software out there which does that. Hot Patch is another one, does the same sort of thing. But in this particular case, you can fine-tune it for different kinds of protein, where these different characteristics of the interface can be given different weights. And on this particular software was used to find a new binding site, and also to predict that these same binding site, allosteric site, would be found in caspases, for example. So looking at the surface is a very useful thing for you to be able to do, and identifying where that surface interacts with something else. Well, normally you have a bit more information. So if you have a lot, even if you only have one crystal structure, if you have a lot of amino acid sequences, then you can combine the information from structure and sequence to try and get useful information. For example, we solved a structure of a superoxide dismutase. It comes from a family of superoxide dismutases, some of which use iron as a cofactor in the active site, others use manganese. But the ligands, the direct ligands, the amino acids which directly hold the metal at the active site, they're the same if it's manganese or if it's iron. But a manganese enzyme won't work with iron in general, and an iron enzyme won't work with manganese. And yet the active sites look almost identical. So how can that be? How can we explain that? So what we did in this case, we did something called a statistical coupling analysis study, where you take an enormous alignment of sequences, preferably more than 1,000 sequences, so that you can see how amino acid residues in different parts of the sequence are coupled together. Like, okay, so if I have a histidine at position 39, the probability is that I'll have an aspartic acid at position 78 or whatever. And it generates a matrix, an enormous matrix of couplings. And what we find is that there are patterns which are different for an iron enzyme and for a manganese enzyme. And these residues, which distinguish iron from manganese, are residues which are close to the active site, but they don't make direct contact with the metal. The idea is that these residues probably do the fine-tuning, which will, at the end of the day, adjust the redox potential of the metal such that it's able to carry out the dismutation reaction itself. So that's an example where you've got lots of sequences and one structure. But it's always better to have more than one structure. So the more structures you have, more information you have. For example, here is an example of a little bit of a structure of a phospholipase. It's a specific phospholipase. It's catalytically inactive, but it's myotoxic. So it destroys muscle via damaging the membrane. And this is the structure, and it's just a bit of the structure I'm showing here, and I'm pointing out one important residue, lysine 122. And this structure we're seeing here is the form of the molecule which has no fatty acid bound to it. These molecules combine fatty acid. No fatty acid bound, lysine is pointing up here. So then we solve a structure with fatty acid bound, and we see that this lysine is pointing inwards. It was the lysine I showed you earlier on, pointing into the structure of the molecule. That's unusual. But that lysine is stabilized by the head group on the fatty acid, which is negatively charged, and the lysine is positively charged. So it looks as though what we can see here is a conformational change in the molecule. When the fatty acid binds, it induces a change in the conformation of this region of the molecule. And that, many years ago, crystallographers would stop at this point because they'd say, well, I've explained it. I've got the form without the fatty acid, with the fatty acid. I can see there's a conformational change, so it's obvious. What happens is that when there's no fatty acid, this lysine is here, sticking upwards. And when there is fatty acid, it moves inwards and uses its positive charge to neutralize the negative charge on the head group of the fatty acid. It's obvious. I don't need to solve any more structures. But if you continue solving more structures, you'll see another red one. So the red ones are with fatty acid bound. And then another one. Oh dear. Now all of a sudden we have one with no fatty acid, which has the structure of one which should have fatty acid. So my nice, beautiful hypothesis about the conformational change is now getting muddy. It's not so easy to understand. So you keep solving more structures. Oh, that's completely different altogether. And another one. And then another one. You just keep solving more and more structures. It becomes a mess. You can't see what's going on until you separate them out. And now what you see is all the red structures are the same, but the green structures are different. Even the structure with the lysine pointing downwards is energetically accessible in the green structures where there's no fatty acid. So what we have is a flexible, it's not a conformational change from two states, 1 to 2. It's a disorganized region of the molecule, a flexible region of the molecule that becomes pinned down in one conformation when the fatty acid head group moves in to the active site. When that happens, phobic residues, the conformational change leads to that fatty acid, that lysine sticking inwards, exposes on the surface of the molecule a hydrophobic cluster. These are all the different structures which have the red conformation. But you see, they all look different. The only thing which is in common about them is they're all hydrophobic and on the surface. That sounds right for inserting into a membrane. So this is the fatty acid in the channel. Okay, this is the lysine pointing inwards, neutralizing the charge. That causes the hydrophobic residues to accumulate on the surface, and then they insert into membranes and cause membrane damage. So I ask, well, why couldn't you see that those residues were particularly important just by looking at the sequences? I don't know how many of you in your bioinformatics look a lot at amino acid sequences, align amino acid sequences, look at the conservation of amino acid residues within an alignment. This normally brings with it lots of useful information. A conserved residue is conserved for a reason. It's doing something structural or functional. Why couldn't we have spotted the importance of the hydrophobic knuckle, which is what starts the damage in the membrane, just by looking at the sequences? Well, the reason is that this is an intrinsically structural motif. If we look at the residues here in black, which form part of the knuckle in each one of these different species of phospholipase, they're not conserved. I mean, not particularly conserved. I mean, there's lots of other bits of the structure which are a lot more conserved than these particular residues. Okay, so it's not easy to recognize that kind of information just by looking at the sequences. We really needed the structural information to be able to answer that particular question. Okay, so I will finish here because I think I've probably spoken long enough. I don't know if there are any questions, but I'd be happy to try and answer them in some kind of a language if you have any questions. But I'll just finish by making a statement about Richard Feynman, the famous Nobel Prize winning North American physicist. He is said to have said, and no one really knows if he did say it or if he said it many times or just once, but what he's supposed to have said is, if you think you understand quantum mechanics, you don't understand quantum mechanics. You really haven't understood it if you think that you're understanding it. It's just too strange to really be understood. So I changed this slightly when I talk about protein structures, which is just to say, if you think you understand protein structure, then you really don't understand protein structure. I personally have been working with protein structure for, well, nearly 40 years, and I'm still surprised by new structures which we solve or which I read about in the literature. There is just so much beauty and elegance and so many surprises in protein structure, which have been crafted over millions of years, tens and hundreds of millions of years by natural selection, that we're always going to be surprised. But we are reaching the point where we now understand enough about protein structure to be able to design new proteins with new folds based on new amino acid sequences. And that's opening up a whole new potential for applications of proteins in biotechnology, even in chemistry, et cetera, et cetera. So we need to fully understand the relationship between structure and function if we intend to apply that knowledge for biotechnological applications, for example. So with that, I will say thank you. I hope you were hearing this. The other day, last week, I tried to give a talk, and I apparently after 15 minutes, the system collapsed, and I went on talking for another 30 minutes, and I didn't know there was nobody hearing anything I was saying. So I hope that's not the case today. And if you do have any questions, I would be more than happy to try and answer them. So I guess what I do now is I click this stop share thing, and we should go back to normal. Thank you all very much for watching. And as I say, if anyone's interested in asking a question, I'd be more than happy to try and answer. Thank you very much.
Thank you very much, dear Dr. Richard. We are so happy for your presentations. In my case, I love structural biology. It's very interesting. Like in my case, interaction between proteins. But we have some questions. Really, thank you so much, dear Doctor. The presentation was amazing. Can you listen to me?
Yes, I can hear you. Yes.
Okay, yes. Thank you. Okay, I'm going to read some questions. There are in the chat box. Okay. The first question is from Juan Martinez. Now, the current potassium vaccines are being proposed. Why we should emphasize the importance to resolve the protein by X-ray crystallography.
Okay. Yes. Well, so what's in a sense, what's the usefulness of knowing the protein structure for vaccine development, for example? Well, there's many potential uses for the structure. Firstly, because you might be able to, if we think about the viral structure, for example, by looking at the structure of the molecules on the surface, you may be able to simplify your vaccine by choosing those parts which will be accessible to the immune system. That's one way in which knowing the structure, knowing which parts of the protein are accessible to the immune system can be used. So you may be able to develop a subunit vaccine where you use just part of the structure rather than using the entire structure of the virus, or the entire bacteria, or whatever, as your vaccine. But also, it can be useful if you know something about resistance. So often, for example, a virus can escape the immune system because it's capable of mutating in certain regions. So it's useful to know where those mutations are occurring, because if that's occurring, that's a mutational hotspot, you probably don't want to be choosing that part of the structure as a basis for your vaccine. So yeah, knowing information about the three-dimensional arrangement of the components of the virus, for example, is extremely useful for the development of vaccine.
Okay, thank you so much for the answer. The next question is from the same person. I think the same person say, is cryo-EM solving the structure of membrane proteins?
not just membrane proteins, but yes, it is solving the structure of membrane proteins, but it's also solving the structure of non-membrane proteins. So cryo-EM is a technique which I'm sure most of you have heard a lot about recently. A couple of years ago, 3 scientists involved in the development of the methodology of cryo-EM, of single particle cryo-EM, won a Nobel Prize. And that's because it's really revolutionizing structural biology. If you look at the rate at which new structures are being deposited into the PDB, then that which is increasing fastest is structures solved by cryo-EM. So yes, membrane proteins, but not just membrane proteins. What are the problems? What are the advantages and disadvantages? So cryo-EM traditionally would produce low resolution models for large particles. Protein crystallography would produce high resolution models, but when particles started to get very big, it was difficult to keep the resolution high. So what's happened over the last few years is with new detectors, principally new detectors, these have allowed, these are electron detectors, these have allowed the correction for movement of the particle during the imaging process. And that correction means that you get rid of the blurring that you would have if it was not corrected. Effectively, you film the molecule, you don't just take a one picture of it. So what that means is that cryo-EM is now able to study molecules which are much smaller than they used to be, down to the size of, I've seen hemoglobin, okay, down to the size of hemoglobin. And the resolution has increased fantastically. So there are now resolutions below 2 angstroms, 1.6 angstroms, for ferritin, for example, something like that. So this is amazing. And if you look at the maps, they're incredible. It's probably not going to be routine for all cases, but it has the advantage that you don't need to form a crystal. But nevertheless, there's a lot of work which has to go into preparing the sample. A good sample is critical. And we are trying to start using cryo-EM, and we are learning. That's a lot of hard work to get a good sample, get a good grid, and then process that to get a good structure. So yes, if anyone here was interested in working in structural biology, I would definitely say the ideal thing nowadays is to know a lot about cryo-EM and something about protein crystallography, because you'll probably need both of them. For example, something where protein crystallography will probably continue to be very important is in the use of structural biology for drug discovery. So for example, in March this year, well, even before March, Chinese groups solved the structure of the main protease of the new coronavirus. And then in March this year, at the synchrotron in Diamond in UK, they were able to screen, I don't remember the exact number, but something like 600 potential small molecule fragments using X-ray diffraction. We can now collect an X-ray diffraction data set in a matter of seconds. So in 24 hours, you can collect literally hundreds and hundreds of data sets. If you have lots of different crystals with lots of different possible compounds, or lots of different compounds possibly bound to them, you can work out what binds where. And with that, in very little time, in about a week or two, they were able to funnel this down to about 60 potential molecules which could be developed into drugs for the development of a pharmaceutical compound against the new coronavirus. And that was all done with protein crystallography. Cryo-EM still doesn't have that potential for high throughput.
Okay, and I suppose that cryo-EM is the revolution for structural protein studies. That's correct or not?
I'm sorry, I didn't catch
that. Cryo-EM is a revolution. It's like the revolution for structural protein studies.
Well, it is. Yes, that's right. I mean, I imagine it's a bit like being back in the 1960s and 70s when protein crystallography was first emerging and people were beginning to see protein structures for the first time. Because for a long time prior to that, people weren't even sure that proteins had a defined structure. Some people believed them to be more colloid-like at the beginning of the last century. With the crystal structures which emerged, it became obvious that we could understand biology at the molecular level, or at least knowing the structures would be an important step forward to understanding biology at the molecular level. And now we're at a similar stage with cryo-EM, because cryo-EM is going to mean not just we have a new technique for solving structures, but that we're going to be able to look at very big, complicated things. We're going to be able to look at complexes, which are difficult to crystallize. Sometimes you can crystallize them, but with cryo-EM, you don't need to crystallize. So we can look at complexes with lots and lots of different components in. And if these structures have different conformations, different molecules in different conformations in the microscope grid, it will be possible to separate the different conformations so you can see how things are moving around. Sometimes we're lucky and we can get some information on that from crystallography. But one of the problems with crystallography is that your molecules are all stuck in a lattice. So the molecules, they're all touching one another in ways which they would not be doing in solution. The advantage of cryo-EM, one of the advantages of cryo-EM, is all of the particles are separate. So there are no distortions present due to the presence of other molecules packed together densely in a crystal lattice. So yeah, there are advantages and disadvantages, but yeah, if you're thinking of going into structural biology, my tip is, first of all, learn cryo-EM.
Great. It's an interesting question. Okay. The next question is, are there software for active sites prediction rather than studying the beta sheets of the secondary structure for the purpose of identifying the active site?
Right. Yes, as I commented, there is software out there which will try to do, yeah, pretty much anything.
Okay.
And there's lots of software which will try to predict an active site. So and they use different approaches. So it could be by analogy or homology, trying to find a molecule which has a similar fold, and then that will tell you where the active site should be. That's, for example, 3D Fun is one example of that. I showed another example where it just tries to find ligand binding sites by comparing with what's in the PDB. So that will give you an idea of where your active site may be, or just for looking for cavities. If your protein has been crystallized without anything bound to it, then just by looking for a cavity is a good hint that you have an active site. The reason that the beta sheet thing works, because the loops following the strands going different ways, the reason that works is because the fact that the loops going in different directions forms a cavity. So you can throw away that. Your structure may not even be an alpha beta protein anyway. Just look for cavities. Okay.
Okay.
Probably the easiest thing is if you have a crystal structure with nothing bound to it. Then if you have an idea of what that molecule does, what reaction is catalyzed by that enzyme, for example, then try to soak into the crystal an analog or an inhibitor, an analog of the substrate. Or if you know an inhibitor, an inhibitor of that enzyme, try and soak that in. And then what you do is you collect the data again with the extra molecule back. And you calculate something called a difference electron density. And this allows you to see, just the electron density, which is due to the molecule which you introduced into the crystal. And that will tell you immediately where your active site is.
Okay, interesting answer. And the last question for time. We don't have a lot of time, dear doctor. And Anders, I'm going to say the last question is, it's very simple. According to Kate Adder, could we trust molecular docking technique to understand protein function, or should we go for molecular dynamics?
Well,
it's an interesting question.
You know, yeah, an interesting question, a difficult one to answer. Yeah, I'm an experimentalist, so I tend to be skeptical about all purely computational based methods. And I'm not criticizing. I'm just saying that we all as scientists have to be skeptical. We have to be skeptical about everything we read. We have to question things. We shouldn't just accept things which are written in the literature. Recently, we corrected a mistake which has been in the literature for 12 years. Very simple thing. But everybody for the last 12 years has been repeatedly saying the same thing, but it's wrong. And it was very difficult for us to convince the referees of our article to publish it. Simply because this wrong information has been in the literature for 12 years. Okay, so you must be skeptical. That's one of the important tools in the scientist's toolkit, is to be skeptical about things, ask questions about things. And so I think you should be skeptical about docking results. You should be skeptical about molecular dynamics. Often with computational studies, if you allow yourself to be fooled, it's easy to be fooled. You can find what you want to find. If you have a preconceived idea of how things should be, it's very easy to see in your molecular dynamics or your docking what you want to see. We're very good at excluding those things which don't fit into our preconceived views, but this is a trap. And it's also something which Richard Feynman talked about. You know, we are the easiest people to fool ourselves, particularly if we're very passionate about a particular hypothesis. We somehow don't see that which doesn't fit with the hypothesis, and we see very clearly that which does. And all kinds of simulation techniques will produce some kind of result at the end of the day. And so we need to be critical in our analysis of those results. Look for other supporting data in the literature, experimental data, which confirm what your simulation is telling you. So the same way I would say that, you know, don't just think protein crystallography or cryo-EM for solving a structure. I don't think you should get married to any particular technique, computational simulation technique. Use the technique which makes most sense for answering the question. So the most important thing, what is the question I want to answer? And then you should be able to choose the best technique. And sometimes you just need to use a mixture of things to get a richer landscape of what's going on.
Okay, great. Thank you so much for your response. Really, I would say both. In my case, I work with drugs, discovery drugs, and I have to use both. First, the docking, and then molecular dynamics. It's very important to both.
Yeah,
work.
very true.
Com os dois, né? com as duas coisas. Okay, thank you so much for your presentation. It was an excellent presentation.
Thank you very much.
You have an email.
It's a pleasure.
If you have an email, you can give us your email for more questions.
Sure. Sure, and I'll just write it down here, shall I? This is my email. I'm writing it into the chat, so if anyone is interested...
In the chat?
In the chat, yeah. So if anyone wants to write to me for whatever reason, then there's my email. I'll be happy to hear from you. And congratulations on this initiative. We're all living in difficult times. I know in Peru, it's been very tough for you guys. We had lots of Peruvian students here. We still have some who haven't been able to get back to Peru. And I hope they will be able soon, and hopefully we'll all be able to get back to life as it was before. So thank you once again for the invitation. It's been great.
No, thank Thank you so much. Muito obrigada, professor, porque você está aqui, sua presencia es muy importante para nosotros. Thank you so much. Muchas gracias. Bueno, a los asistentes, recordarles que esta serie de webinars es organizada por la Sociedad Peruana de Bioinformática, ¿ok? Y por la Comunidad Científica Lambayecana. Agradecemos nuevamente al doctor Richar por su interesante presentación. Thank you so much for your interesting presentation, Professor, and for your support in this event. And the diffusion of the science is very important, your presence, and the best wishes for you in your work and your future. Okay. También agradecemos a los asistentes por su apoyo. Es por ustedes que también realizamos eventos de divulgación científica, de difusión científica en bioinformática y biología computacional. Esperemos que continúen en los siguientes webinars que hemos programado. El próximo será este viernes, el viernes 22 a las 6 de la tarde. Y por favor atentos a nuestras redes sociales y cualquier consulta, ahí está nuestro correo, contacto@bioinformatica.pe. Bueno, muchas gracias a todos. Thank you so much for your presence today. And with me, we'll be in the next time. Thank you so much, Professor, and thank you so much for everyone. Bye bye.